Rab-GTPases control membrane trafficking events throughout the cell. Mutations in Rab18 cause the neurodevelopmental disorder Warburg Micro Syndrome, but the precise cellular function of this Rab protein is unknown. Warburg Micro Syndrome is also linked to mutations in the Rab3 gap complex, which inactivates the Rab family member Rab3. But Francis Barr from the University of Oxford wondered whether the Rab3 gap complex might also interact with Rab18.
Patients with mutations in Rab18 or either of the two Rab3 gap subunits have the same disorder. So you would say, well, Rab18 and these components are acting in the same pathway. So we started looking at what would this Rab3 gap complex do and how might that relate to Rab18 function.
Working in collaboration with Irene Alianis and Mark Handley, who previously mapped the disease-causing mutations in Rab18 and Rab3 gap, Barr and colleagues, led by Andreas Gerondopoulos, reasoned that the Rab3 gap complex could either be a downstream effector of Rab18 or else be an upstream regulator of the small GTPase, serving as a guanine nucleotide exchange factor, or GEF, to activate Rab18 and recruit it to a specific cellular membrane. The researchers found that the Rab3 gap complex specifically stimulated nucleotide exchange by Rab18 in vitro, and that ectopically targeting Rab3 gap to mitochondria was sufficient to recruit Rab18 to the outer mitochondrial membrane.
And we could take the wild-type complex or complexes which carry the disease-causing mutations, and obviously the clear prediction there is that disease mutations should not trigger nucleotide exchange and don't promote membrane targeting, and that's what we found, that they don't. So we had very strong correlation there between the biochemical activity of the complex, the membrane targeting activity, and the disease.
In contrast, disease-causing mutations didn't impair the Rab3 gap complex's ability to inactivate Rab3, suggesting that defects in the activation and recruitment of Rab18 underlie Warburg Micro Syndrome. But where do Rab3 gap and Rab18 normally localize?
We could show that Rab18 is distributed throughout the ER, and the GEF complex seems to localize to subdomains of the ER tubular networks. So it's very much in the right place for something driving recruitment of Rab18 to the surface of the ER and potentially controlling ER function.
Indeed, knocking down the Rab3 gap complex caused Rab18 to relocalize from the endoplasmic reticulum to the cytosol.
So the ER is really in fine balance between extended tubular networks pushing out into the cell periphery and sheets of ER membrane close to the nucleus. Those structures look wildly different, but they're in fact connected together. They're all part of the same thing. And what we can see is if you take away Rab18 or Rab3 gap, the balance of ER tubules to sheets is altered. So we lose the organized tubular networks and we have an enormous spread of ER sheets. So they kind of take over the structure.
The expansion of ER sheets in the absence of Rab18 could be reversed by re-expressing the small GTPase. Moreover, cells from Warburg Micro Syndrome patients showed similar defects in ER morphology, with ER sheet volumes increased at the expense of ER tubules. Rab18 has previously been suggested to regulate the formation of lipid droplets, so Gerondopoulos et al. Looked to see what happens to these organelles in the absence of Rab18 or its exchange factor Rab3 gap.
So the size of lipid droplets increases, and that seems to be in proportion to the increase in sheet ER. So one possibility is simply by increasing the surface area of the sheet ER, the lipid can be more efficiently transported into lipid droplets, and they simply get larger as a consequence. So that's something that would be a very real physiological consequence of interfering with this pathway, but would potentially be a kind of indirect cell biological consequence arising from altered ER structure or trafficking at the ER.
However, Barr and colleagues think that Rab18 and Rab3 gap's direct function in shaping the ER may be important for neuronal function. Indeed, mutations in several other proteins that control ER morphology, including spastin, reticulum 2, and the atlastin GTPases, have been linked to hereditary spastic paraplegia, a disease with similar symptoms to Warburg Micro Syndrome.
At the moment, our thinking is very much that this is a pathway which is important for ER dynamics. So the ability of the endoplasmic reticulum to spread, for example, down axons and dendrites, may be compromised if you have a more rigid or static sheet structure. And if you lose the tubular networks, the ability of ER to kind of colonize axons and dendrites may be reduced. And if you don't have localized ER, then you have some problem. So I think the two major questions are, how does Rab18 exert its functions? So you activate Rab18 on the surface of the ER. What protein complexes get recruited? What functionality have you brought in? And why does that have a consequence? The slightly longer goal, but I think actually in some ways the most important thing, is to extend the cell biology into disease models, really to start looking in neuronal cells and animal models, what happens during development to the endoplasmic reticulum in the normal situation and how that is really altered in Rab18 and GEF complex mutant animals.
In the meantime, however, you can learn more about how Rab18 and its exchange factor Rab3-GAP control ER structure in the paper by Gerondopoulos et al., published in the June 9, 2014 issue of the Journal of Cell Biology.